You all know about bile juice. Bile juice plays very important role in emulsification of food, and also it helps in the production of proteins and cholesterol. And you all know about glycogen. Glycogen is stored in the liver, and also it controls various metabolic processes of carbohydrate metabolism, protein metabolism, and also lipid metabolism. And it has many other functions. And liver undergoes various diseases, mainly liver cancer or HCC. And there are many risk factors for chronic liver diseases, including hepatitis B virus, hepatitis C virus, and also excess alcohol consumption and aflatoxin B. And also nowadays we consume high amount of junk food, and also after year 2000 we all have sedentary lifestyle. Dr. Keshav Prasad know very well about sedentary lifestyle in IT field. He was working in Whitefield in Bangalore, and this is becoming very common not only in Bangalore, across the globe. And also nicotine. Dr. Tapas Kundu talked about harmful effect of Kadipudi and tobacco. And nicotine is the main compound. It's also indirectly associated with liver inflammation and liver diseases. And there are many environmental pollutants, including air pollution. And also nowadays we have mental health issues and also epigenetics. All these factors play major role in controlling various chronic diseases. And if you go to USA, like if you see a lot of people, not only white people, even black people, too much obese. These are like morbid obesity is very common. And my weight was only 64 kilo before going to USA. Once I came back from USA, my body weight was 94 kilograms. Now I'm 84. And this is due to like sedentary lifestyle and consumption of Western diet and all those things. And it is directly related to non-alcoholic fatty liver diseases. Now we call metabolic associated liver diseases. And long-term progression of this fatty liver and metabolic non-alcoholic fatty liver diseases will become liver cancer. And global prevalence of MSLD varies with different countries. Nowadays, due to globalization, westernization, everywhere MSLD is becoming very, very common. And also, prevalence of NFLD is directly associated with HCC. NFLD is becoming one of the major contributing factors for liver cancer or hepatocellular carcinoma. And prevalence of MSLD in India is also varies from 9 to 53%. According to recent report from Zydus, a company in Gujarat, they mentioned that almost 400 million Indians are suffering from fatty liver without knowing it. 400 million means almost 40 crore people in India, we have fatty liver disease. And also coastal South India has 49.8%, and urban communities in North India 53.7%. Urban population is due to the consumption of excess food. Nowadays, affordability to have food is very, very easy, and that is also one of the major contributing factors. And also rural community in North India is 30.2%. And if you see, like we have normal liver, healthy liver. If you feed on high fat diet, high calorie diet, high glucose, high fructose diet, this liver will start accumulating too much fat. That we call fatty liver. If you have fatty liver for 5 years or 10 years, it will slowly becoming inflamed liver. That we call hepatitis or non-alcoholic hepatitis. Like after 5 years or 10 years, this inflammation will lead to fibrosis, cirrhosis, and finally people may have liver cancer. Once if you have a later stage of fibrosis, liver will lose its function, then there is only one option is to have liver transplantation. Otherwise people will die. And if you have small tumor liver, we can cut that, we can remove that tumor. But if you have cirrhosis, it is very dangerous thing, and it is somewhat deadly. Like after some time, people may get liver cancer. If you have too much cancer in your liver, we need to remove that liver. We need to go for transplantation. And there are many pathways are involved in liver cancer. majorly like receptor tyrosine kinases pathway, RAS pathways, MEK, RKF pathway, and PI3K, AKT, FR pathway, and Wnt beta ketamine, JAK-STAT pathway, and EPO pathway. These are all common cancer-associated pathways, and these pathways plays very important role in immunoevasion, stem mass, angiogenesis, metastasis, and even they play very important role in metabolism. mTOR, AKT pathways, they play very important role in metabolism and also proliferation of these cancer cells. And quite some years back, we showed that even three of the major components of RNA-induced risk complex also plays very important role in hepatocellular carcinoma, where like AG1, SND1, AGO2, these are three important components of risk complex. These components are also overexpressed in liver cancer. Based on that, we published a paper in hepatology, and some of you know how this microRNA works from yesterday. We are hearing from many people, and yesterday Dr. Praveen Kumar Shetty and Dr. Sanjeevan, they presented a lot of data about microRNA and risk-related proteins. And this is how microRNA biogenesis takes place inside the nucleus, and they undergo processing with the help of TROSA and the DGCR8 protein, and they will export it into cytoplasm with exporting 5 protein. Once they come to cytoplasm, these microRNA undergoes further modification with the dicer, and once they become single-stranded RNA, they align with target mRNA of various genes. If they have perfect alignment, and they will go to risk complex. This risk complex has two enzymes, ribonuclease enzyme, SND1 and AGO2. These enzymes, they finally chop this mRNA, like mRNA microRNA complex, and they will degrade so that they will replace the expression of protein synthesis one side. In another side, if there is no perfect alignment, imperfect complementary also stops translation. This is how this microRNA risk complex suppresses or represses various protein synthesis by degrading mRNA or stopping translation. This is one type of epigenetic regulation. This is taking place after the transcription. This we call post-transcriptional gene regulation through risk complex and with the help of microRNAs. And these two Nobel laureates, Craig Mello and Andrew Fire, they discovered RNA interference technology, and they got Nobel Prize in 2006. And for their discovery of RNA interference, they got this Nobel Prize. And this year, like last year, year 2024, Victor Andrews, Gary Wickman, they got Nobel Prize for the discovery of microRNA. This discovery, microRNA discovery and RNA interference discovery, led further to elucidate the enzymes which are present in this complex. This Victor Andrews and Gary Wickman and even Craig Mello and they didn't mention which are the enzymes which are involved in RNA interference, but later Professor Gregory Hammond discovered this SLE1 and AGO2 enzymes, and he showed complete mechanism of this complex. Later in the year 2010, my mentor Dr. Devanand Sarkar, along with one of my colleagues, we showed this AG1 protein is also interacts with this risk complex, and it plays very important role in risk machinery. 2 years back, somebody shown that if you disrupt SND1 AG1 interaction, you can prevent breast cancer. Like that, they published paper in Nature Medicine and Nature Review of Cancer. And these are the 3 important proteins: arginine 2, SND1, and AG1. These are overexpressed in liver cancer. They play a very important role in proliferation, migration, invasion, and also tumor angiogenesis, and also they play a very important role in risk activity, and also they play a very important role in hepatocarcinogenesis and adhesion and tumor metastasis. And microRNA associated risk machinery is a novel target for SCC. Based on that, our own study, we propose some hypothesis where microRNA regulates pre-current post-transcription gene expression through risk machinery, and also risk components SND1, AGO2, AG1 are overexpressed in SCC, and therefore targeting these risk proteins by natural compounds has potential importance. Why natural compounds? Until now, you heard about toxic effect of various drugs. There are a lot of side effects, including sodium valproate and even metformin, all these drugs, they have a lot of side effects, and even sorafenib, well-known compound which is giving for liver cancer, that is also has major toxic effect. And what is the need for our study? Virtually, there are no effective treatment for MSLD and advanced symptoms of MSLD, death, liver cirrhosis, and HCC. Therefore, identification of novel targets to develop an effective treatment strategies using natural compound is need of the hour. And why natural compounds? Dietary natural compounds such as phytochemicals play a beneficial role beyond basic nutrition. And the beneficial effects of these dietary bioactive compounds mainly rely on their influence on the transcription factors and their ability to inhibit the obesity-induced MFLD and HCC. And we screen many compounds, and by screening, after we finally choose a compound called capsaicin. And this is our overall hypothesis in this study. One of my PhD students, she chose two different compounds. One is capsaicin and another one metformin. And now currently I am presenting my data which is capsaicin. And capsaicin is an active component of chili peppers. You all know about that. And which are, chili pepper is a plant belong into the genus capsicum, and it has IUP is named like 8-methyl 6 nanoenamide, and this is molecular formula of capsaicin. Molecular weight is 3.305.4, and this is the structure of chili, and it has various beneficial effects, and it acts as anti-obesity, antioxidant, analgesic, and it plays a very important role in urinary disorders, and it helps. It has anti-cancer effects and antimicrobial activity. And you all know this is the published image online. This is one side you can see this diet rich with fruits, vegetables, and also low calorie diet. In another side you can see diet which is having high calorie. And here you can see the picture of liver, and you can see the picture of, you will come to know about overall my study. And we used three different cell lines. One is 3T3L1 cell line, and another one is HT2 and R7. This 3T3L1 cell line is the adipocyte cell line, mouse adipocyte cell line. We usually use this cell line for obesity or adipogenesis related studies. And this HT2 is the epitoma cell line, but this is not aggressive cell line. It will not form tumor in mice. And we use this cell line as almost normal cell line. And this is R7 cell line, which is isolated from one Japanese patient. It is a highly aggressive cell line. It behaves like as cancer cell. And this is like, here we use 3T3L1 mice adipocyte cell line, and we use a differentiation media. And when we use differentiation media with fatty acid, there is accumulation of these like lipid droplets inside the cell. And after the treatment of capsaicin, you can see downregulation, this less lipid accumulation after the dose-dependent treatment of capsaicin. Here it shows this capsaicin prevents lipid accumulation in 3T3L1 cell line. 3T3L1 cell line is the adipocyte cell line. We all know about adipose tissue. Adipose tissue is the major storage area of fat. This is how capsaicin prevents this lipid droplet deposition. And later we did adipokine array. Adipokine array is a commercial kit array. Here we get four different membranes. These membranes are pre-labeled antibodies. Whenever we treated cells with capsaicin and we collected this conditioned media from the cell line, we put this conditioned media which was collected from treatment and control, and we got this result. This is in control membrane, this is in capsaicin treated membrane. This capsaicin treated membrane contains conditioned media from treated cells. This is untreated cell. You can see there is a downregulation of various cytokines, adipokines, and you can see IGF-PP3 is going down, IGF-PP6 and HGF and lipocalin-2, but you can see there is slightly increase in adiponectin. Adiponectin is a good adipokine and which prevents obesity and also various other metabolic syndrome related things. And this is our experimental setup. We used Swiss albino mice, and these Swiss albino mice were divided into different, 4 different groups, 5 different groups, and we treated with Western diet and sugar water. Sugar water contained fructose and glucose solution. After 8 weeks feeding, mouse become somewhat obese, and after 8 weeks after when they become obese, we started treating with capsaicin for 4, 8, and 12 weeks. This is the result we got. You can see this is in 12 weeks mice, 16 weeks, 20 weeks mice. You can see when we gave Western diet, you can see accumulation of too much fat. After the treatment, you can see reduction of fat mass. This is in control mice. And we also measured the body weight and also fat mass, and also we did ITT and GTT assay to check the insulin resistance in these mice. And we found that after the treatment of capsaicin, body weight automatically reduces, and also fat mass reduces, and also there is a less insulin resistance when we treated with capsaicin. This is the liver picture. You can see these are control liver picture. These are fatty liver picture, and you can see after the treatment of capsaicin, they look like this. By just observing their color, we can tell that this is fatty liver. These are normal liver, and we measured liver weight and even liver enzyme. Liver weight goes down after the treatment of capsaicin, and also all these liver enzymes which are upregulated in fatty liver diseases and HCC, after the treatment of capsaicin, they got down. And this is HDL, and we measured various lipid profile after the treatment of capsaicin, and HDL goes up after the treatment of capsaicin, but LDL and cholesterol and also triglycerides, they go down after the treatment of these capsaicin in mice. And we did the same thing with in vitro using cell lines T2 and R7. When we treated these cells with palmitate and foliate, we found that there is a more accumulation of this lipid droplets inside the HT2 cells. After the treatment of capsaicin, there is less accumulation of lipid droplets. Same thing with R7 cell line, another cell line. And this we did oil redo staining in mouse liver. This is in vivo, and we found same thing with mouse also. This shows both in vitro, in vivo accumulation of lipid droplets inside the liver was downregulated after the treatment of capsaicin, and also decreased fat mass. This shows capsaicin inhibits obesity and also fatty liver. And we measured some of the fatty acid synthesis markers, SRB12C, and also fatty acid synthesis genes, both in vivo and in vitro. We found that these markers got downregulated after the treatment of capsaicin. This later we did HND staining, and we found that you can see here 12 weeks, 16 weeks, 20 weeks mice liver image, and here these are control mice. You can see here fat accumulation in the liver, and also after the treatment of capsaicin, they almost look like normal liver. And we also measured inflammation.